Hello, welcome. Thanks for joining us for another research and development update. Today, as promised, we're going to focus on our multi-targeted cancer program. I'm Kevin Kircher. I'm the Communications Director for Ocean Biomedical, and we are joined here today by our scientific co-founder, Dr. Jack Elias, who is also the chair of Ocean Biomedical's Scientific Advisory Board. Welcome.
Thank you. Thank you. It's a pleasure to be here.
It's great to have you. Dr. Elias is an internationally renowned pulmonary researcher and clinician who's done a lot of work in this space. I know you've developed even some of the mouse models that are used globally to study lung diseases, and certainly worked in a number of other areas for pulmonary research. And Dr. Elias is the past president of the Association of American Physicians. He served for a time as the physician-in-chief at Yale New Haven Hospital, and he recently retired as the Dean of Medicine and Biological Sciences here at Brown University in Providence. And I know that those are just a few of your titles you've held over the years.
Thank you.
We want to take a closer look at Ocean's cancer program today, and which is developing multiple cutting-edge cancer immunotherapy approaches. Almost all of them, or all of them, focus on regulating chitinase 3 like 1. And we've characterized that as a master switch in the cancer process. And I want to we'll spend a little time today getting to understand what chitinase 1 is, what chitinase 3 like 1 is, and why it's such a powerful target for us, and why Ocean is putting so much into that effort. Dr. Elias's cancer discoveries in this space, we have an antigen we call FRG. It's anti-chitinase 3 like 1.
It's an antibody.
Antibody, thank you. First of probably several mistakes today. I'll do my best. But it has received multiple U.S. and international patents, and they apply for potential use in lung cancer, brain cancer, in melanoma, in prostate cancer, in colon and rectal cancer, kidney cancer, and in ovarian and breast cancer. And just that list alone is phenomenally impressive. And I want to start back with the idea of what is chitinase 3 like 1? And why is it such a powerful operator in this space? So how did you get on to chitinase 3 like 1?
It's a very good question. We stumbled. We have to be lucky in science as well as smart in science. And we were lucky in that we stumbled into chitinase 3 like 1. We were studying asthma at the time, and we realized that one of the major things that goes on in people who have asthma is that they have what's called a type 2 immune response. We then realized that this molecule, chitinase 3 like 1, which also is called YKL40, it's also called CHIL1, it's got a lot of other names, but for the sake of today, we'll call it chitinase 3 like 1, turns out to be a major driver of that whole process. And we were very interested in the finding that the levels of it in the circulation were increased in people with asthma. We eventually wrote that up. We sent it to a prominent journal to get published. And one of the criticisms from the reviewers was, is this moiety, is this chitinase 3 like 1 increased in just asthma, or is it increased in other diseases as well? And so we said, well, that's an easy thing to do. So we went to the clinic and we got serum samples from healthy people and from people with a whole variety of diseases. And it turns out that the highest levels we saw were actually in people with cancer. And that sort of led us to the realization that chitinase 3 like 1 might play a major role in the pathogenesis of cancer. And that started us down a multi-year pathway that has now left us with the understanding that chitinase 3 like 1 is a truly important and powerful regulator of multiple oncogenic or cancer-causing biologic pathways.
Your research and development team has been working on monospecific approaches and also bispecific approaches to several different cancers. And I want to know, first of all, can you tell us about the anti-chitinase 3 like 1 mono antibody
So the first idea for a therapeutic was to develop an antibody that could neutralize the effects of chitinase 3-like-1 by itself. That's the monospecific antibody that you're referring to, Kevin. And it's turned out to be tremendously powerful in a whole variety of diseases. So it's not just cancers, it's powerful in other diseases, including fibrotic diseases and other settings. And so we have a moiety which is very, very powerful in terms of its ability to control chitinase 3-like-1. We then realized that we might even be more powerful if we added to that the ability to simultaneously target other targets. And one of the things that we got very interested in that we can talk more about if it fits for today is that we think that chitinase 3-like-1 is a master regulator of immune checkpoint inhibitors. Immune checkpoint inhibitors are the pathways that control the host innate immune response against cancers and other diseases. And when you control chitinase 3-like-1 itself and you simultaneously control via a bispecific antibody, when you control the immune checkpoint inhibitors, you get a really tremendously powerful effect in the diseases you're studying, in particular in non-small cell lung cancer and glioblastoma multiforme.
Before we move on, let's just give us a clear sense of what is chitinase 3-like-1 doing when it's operating around the cancer, and why does the anti-chitinase 3-like-1 seem to work as well as it does?
Well, chitinase 3-like-1 is a moiety that, first off, we all have it in our blood. Every one of us, when we're healthy and when we're not healthy, you have it in your blood all the time. What happens is when you get sick, whether you get infected, you get inflammation of some sort, you get remodeling diseases like emphysema and COPD, for example, or in fibrosis, the levels go up. In cancer, chitinase 3-like-1 is induced, and it is tremendously important in terms of stimulating the pathways that cause the cancer, and it's tremendously important in stimulating the pathways that make the cancer progress and get worse. There are at least 7 different pathways that chitinase 3-like-1 regulates. So there's 7 different pathways that anti-chitinase 3-like-1, our antibody, controls as well. And then when you add in the bispecifics, you have this tremendous ability to simultaneously control multiple different cancer pathways with the same single intervention.
Wow. We had a graph, we put it up a couple times already, showing how the anti-chitinase 3-like-1 affects mouse lungs. And how much of a difference did you see? How much of a difference did it make when you applied that just in the mouse models?
Well, it depends on which model you're talking about and in what setting, but the two models we use most frequently, one is a model of what's called metastasis to the lung. Here in this model, the animals are given malignant melanocytes, malignant melanoma cells, and the malignant melanoma cells spread to the lung over time, and they form actual nodules on the surface of the lung. And you can easily see in that setting a 80–90% decrease in tumor burden if you use our antibody compared to the control antibody. The other techniques that we use are techniques where the mice are genetically modified so that they have the same genetic mutations that people have when people develop non-small cell lung cancer. And we're talking in particular mutations of the KRAS oncogene, or P53, and those mice develop primary cancer. They also are decreased in terms of tumor burden up to the tune of 80–90% when you give them our antibody.
Fascinating. You had 2 times this year, there was major news that has kind of spun out of this on the glioblastoma front and in terms of non-small cell lung cancer. How, and both involve some independent researchers, adopting your ideas and putting them into play. How big a news was this in your career? All the work that you've done, how big a news was this for you to get the results from Nikos Tapinos' lab and and on the non-small cell lung cancer?
What I can honestly say is that the findings in malignant diseases, including non-small cell lung cancer and glioblastoma multiforme, are among the most impressive findings in my almost 4 decades of cancer research and other forms of pulmonary research. It's really very exciting when you have a disease like glioblastoma multiforme, which is a tremendously aggressive form of brain cancer, which really has no really effective therapeutic intervention. It's very exciting to have a human model in the mouse and then to have a therapeutic that actually works to control that model to the degree that ours is doing it, and to actually understand the mechanism of it. One of the things that's very characteristic of glioblastoma multiforme is that it spreads very rapidly. And if you look at the cancer cells in patients with glioblastoma multiforme, some are neuron-like and some are what are called mesenchymal. If you have the mesenchymal form of glioblastoma multiforme, that's the worst prognosis that you can have. And what we now know is that chitinase 3-like 1 drives the cells in a mesenchymal direction. This is work, as you said, from Dr. Tapinos' laboratory using our antibody. And if you give them our antibody, you block the progression to the bad phenotype of the disease. In non-small cell lung cancer, we have also made tremendously important new observations. If you want, I can go into those in detail as well.
Let's look a little bit closer at the glioblastoma findings first. And so number one, Dr. Tapinos found out that chitinase 3-like 1 actually not only has a role in glioblastoma, which was not known before, I believe, but has a role in creating the more aggressive strains.
That's right. It drives it in a mesenchymal differentiation progression. And so you have the tumor becoming mesenchymal in terms of the proteins that it puts on its surface, and those are the tumors that have the worst prognosis. And our antibody is able to block the chitinase 3-like 1, which is the driver of that process.
That must have been amazing to have.
Very exciting.
I think we have a graph that shows some of the results. Do you remember when you applied the anti-chitinase 3-like 1?
Well, in terms of the differentiation in a mesenchymal direction, you're talking 70–80% decrease in differentiation toward the bad phenotype. And when you talk about the growth of the tumor itself, in terms of just tumor burden, you're talking 80% or so decrease in tumor burden based on the administration of our antibody.
Amazing, amazing. Similarly, in the non-small cell lung cancer space, tell me about the EGFR mutant. And it sounds like the same thing happened, and we just announced this a month ago, but realizing for the first time that chitinase 3-like 1 has a role in this prominent EGFR mutation.
So first off, when you talk about lung cancer and you talk about EGFR mutant lung cancer, what you're talking about is a group of diseases that are called non-small cell lung cancer. 85% of them are related in some fashion. They all have driver mutations. And one of the most common driver mutations is this mutation in EGFR, which is the epidermal growth factor receptor. This occurs in about 20–25% of people with a European heritage. It occurs in 40–50% of people with an Asian heritage, and people do not understand why it's more common in people with an Asian genetic background than a European genetic background. But what is very, very clear now is that the mutations in EGFR drive the production of chitinase 3-like 1. Chitinase 3-like 1 feeds back to drive the activation of EGFR, and that's the reason why these diseases grow and progress. And we now know that our antibody blocks that entire process and blocks the development of EGFR mutant cancers, in particular ones that have the standard mutations in this disease. So it's tremendously exciting that our antibody is able to have this tremendously important effect on EGFR mutant non-small cell lung cancer.
My sense was from reading that paper is that in this case it not only blocked progression, but it actually reduced the tumors, existing tumors. Was that correct?
Yes. Yeah, it blocks progression, and by blocking progression stops them from growing in terms of size. The other thing is that it modifies whether people respond or don't respond in an appropriate fashion to the therapies that are already out there for EGFR mutant non-small cell lung cancer. So the standard of treatment right now for people with EGFR mutant non-small cell lung cancer are a class of drugs called tyrosine kinase inhibitors, or TKIs. There are 3 generations of TKIs that have been studied and approved by the FDA. The 3rd generation is now the sort of standard of care. The most commonly used one is a drug called osimertinib. And people with these EGFR mutations respond very nicely initially to osimertinib, but somewhere between 10 and 18 months after they start the drug, they stop responding, and then the tumor comes back and the tumor progresses and the tumor spreads or metastasizes. We showed that the tumor cells that are resistant to osimertinib become sensitive to osimertinib if you give them our antibody at the same time. So our antibody restores the therapeutic sensitivity to osimertinib in this setting, which is a very exciting finding.
We have a question about the market potential for the cancer programs, and I know that there was some discussion when this paper came out about the existing market for the TKI inhibitors and what it might mean to be able to extend their therapeutic life, if that's possible. Do you have any insights on that?
No, I leave that to the bean counters.
How big a need, though, can we say at least, how big a need is the non-small cell lung cancer space?
The non-small cell lung cancer space is a very, very big space. You're talking 450 to 500,000 people, and you're talking a good 50% of people with non-small cell lung cancer present with a disease that's advanced enough that surgical therapy is no longer going to give you the results that you want, and you have to go to some form of chemotherapy or immunotherapy. And this is where our antibody and osimertinib and other tyrosine kinase inhibitors all work together in a really exciting way. It's exciting that people respond to osimertinib initially, but it's even more exciting that by giving our antibody, you can prolong and solidify that response for an even longer period of time.
We shared a slide that shows what happens when you apply just our antibody, what happens when just osimertinib, and then what happens when you do the 2 in combination. Do you want to say anything about that? And it seems like the combination results were, aside from the resensitization, even before that they work very nicely together.
They work very, very nicely together. Probably the simplest way of thinking about it is one of the things you want your tyrosine kinase inhibitor or your antibody to do is to induce the death of tumor cells. The death process that's being induced is called apoptosis. It's a programmed cell death process. And it's very clear that once the cells develop resistance to osimertinib, osimertinib does not cause the cells to undergo apoptosis. But if you give our antibody, 40 to 50% of the cells die right off the bat, which tells you that you have restored the ability to induce the death of these tumor cells, which is exactly what you want to do therapeutically.
And this is often put under the title of cancer immunotherapy, but it's actually allowing the body's own immune system to operate. Well,
the cancer immunotherapy is related to the immune checkpoint inhibitor pathways that we talked about earlier. The mechanisms of how we restore sensitivity to osimertinib is probably not mediated through that pathway. But what we do see is when you do that, you induce what are called tumor suppressor genes. So you induce P53, you induce retinoblastoma, you induce a tumor suppressor gene called P10, and you induce a tumor suppressor gene called KEAP1. And these are the genes that suppress tumors normally, which are tremendously important in keeping tumors under control. The effect of inducing them is that you now can control the tumor to a much greater degree than you could before.
Interesting. You've called chitinase 3-like-1 a master switch in cancers.
Right.
We had this big glioblastoma paper came out in April. The non-small cell lung cancer just came out this past month. Do these buoy your confidence in that claim?
We're tremendously excited by the findings. It's, you know, one of the things that oncologists have known for a long time is that to really have a maximally effective therapy for cancer, you want to not just have a single therapy, you want to have multiple therapies that all attack the tumor in slightly different ways, so that if one stops working, the other one will still work. And that's why for many, many years, we used what was called combination chemotherapy. And that's why when you talk to friends who, or you have relatives who underwent cancer therapy, they would get combinations of drugs that are put together to try to maximize the effects on the tumor. We now have the ability to say with certainty that the antibody against chitinase 3-like-1 attacks 7 different oncogenic pathways simultaneously. And that's why we think, at least that's why we're convinced that it's so effective in every setting that we've tested it in.
Tell me about that. Expand on that. It's not just one cancer that you're targeting. It's not even just one cancer pathway you're targeting. You're actually targeting multiple pathways within multiple cancers.
You're simultaneously targeting multiple pathways. So you're targeting the apoptosis pathways that we just talked about. You're targeting all the different immune checkpoint inhibitor pathways. So they're talking PD1, PDL1. We're talking the CTLA4, B7-1, B7-2 pathway. We're talking about LAG3 and others. You also have a situation where you're targeting coughlin. You have a situation where you're targeting the BRAF proto-oncogene. There is now data that the ability of neutrophils to produce nets and control T cells, apposition to tumor cells and killing of tumor cells is also mediated by chitinase 3-like-1.
All right, and that is not something we've talked about at all before, correct?
That's a very recent finding from colleagues in Canada at McGill University.
We had a question here that came in, any other big news on the horizon, and it does sound like having discovered that chitinase 3-like-1 is a master switch in the cancer process, now you and others are going out and just expanding the understanding step by step of how that operates. And it's very exciting that so many different pathways are able to be targeted. And once you know that, you can say, okay, where are these pathways operational? In what diseases and at what stages of these diseases are they operational? And once you have made that, once you have that information, you say, okay, now it's time to test what our antibody does in that setting. We've been talking a lot about the deep research. These assets are preclinical right now. A couple folks have asked about what is the process, what is the process from here? What are the steps you need to take to actually get these in the hands of doctors and into patients? So
we are at a stage where we are working with colleagues in various research settings to do the studies and to get the data that's required to get IND status. IND stands for Investigational New Drug status. We are hoping in 12 to 18 months we will be IND approved for our antibodies. And then once that happens, we move into phase 1 studies in people.
What do you think, has any one of these particular applications been designated as the first to really target? I know you
Well,
haven't. And what do you think should be?
what you're saying, in my opinion, the thing that I would suggest we target first are people who have developed resistance to osimertinib, who have non-small cell lung cancer. If you have non-small cell lung cancer, you've been treated with osimertinib, you responded initially, but you're now not responding as well or at all. Those are the people that I think would be the first people that we should test in terms of the efficacy of our intervention, and see if what we see in the experimental settings is actually happening in people. And if it is, it will really be a tremendous improvement for those individuals. So that would be called salvage therapy for people with EGFR mutant non-small cell lung cancer who have therapeutic resistance to osimertinib.
Amazing. I want to make sure we've covered, we talked a lot about the monospecific antibody. We did not talk very much about bispecifics that you are developing. What is the power of the bispecific antibody, and what is the potential there? And do you want to say anything about any of the ones that Ocean is working on currently?
So the exciting thing about bispecific antibodies is that you can add two different antibodies that you would normally use individually and put them into the same molecule, so that that same molecule simultaneously targets both of the targets. We now have bispecific antibodies that target chitinase 3-like 1 and at the same time target PD-1, which is one of the most important immune checkpoint inhibitors. And we have another antibody which has a target of chitinase 3-like 1 and CTLA-4, which is also an immune checkpoint inhibitor. When you do them in combination, you get a level of power and a level of strength that you don't get from the individual moieties. And it's truly tremendously exciting to see the results of this. And there's a whole series of diseases you would then want to take them into, diseases that either respond to chitinase 3-like 1 by itself, or respond to PD-1 by itself, or CTLA-4 by itself. But when you put them together, you can have a tremendously extended and more powerful response. And so we're very excited about moving in that direction as well.
That's wonderful. So it really does sound like there are multiple channels almost that this cancer program is following, and each of those can split into multiple cancers. You know, I'm seeing this question about market potential. Have you ever added up all the market potential?
I've never done it. That's what you need KPMG or somebody else like that to do. But when we did talk this through with KPMG, when we initially formed Ocean, they were very, very excited about the possible effects of the antibody by itself and the bispecific antibodies. And we are very much very excited as well. The fact that this is, we're focusing on glioblastoma, multiforme, and non-small cell lung cancer. But as you accurately pointed out, there's every reason to believe that breast cancer, pancreatic cancer, prostate cancer, and a variety of other cancers will respond to this intervention as well.
Amazing. Someone has also mentioned the recent partnership with Virion Therapeutics, and we've just added a fourth program to Ocean Biomedical with their hepatitis B research, which is moving forward as we speak. Very exciting. And I think most people probably haven't noticed yet, although it was in some of the announcements, that they have a cancer therapeutic as well. The hepatitis B program for them is under the name of Viron 0200, and next up is their cancer therapeutic, which is Viron 0300. And I think maybe at a future date, it might be fun to have the folks from Virion in here to talk a little bit more in detail about that program. But I know, just to close, let me see if there's no other questions coming up, but just to close, I know that you have dedicated a huge portion of your professional career to research, advancing research on your own, and as an administrator to advancing translational research more broadly. and to making it more possible to get great discoveries, to move them through the development stage into real medicines that can help people. And I just wanted to know if you want to say anything about that and your commitment to that and the commitment of Ocean Biomedical to that process.
Well, thank you. I'm happy to comment on that. I've spent most of my professional life trying to combine research that answers and that asks the appropriate questions and answers the appropriate questions, and the teaching of students, whether they're undergraduate students, graduate students, medical students, interns, residents, and so on. One of the saddest things that can happen in a life of a patient is to go to their doctor, have a problem, have a diagnosis be made, and be told that there's no therapy for what you have. One of the saddest things for a physician is to diagnose a patient and then have to look the patient in the eye and say, we don't have a good treatment for that. That's why I've dedicated the vast majority of my life to asking the right questions, getting the right answers, and then developing the new therapeutics and the demonstration to the next generation of physicians and scientists of the power of that approach. I'm actually thrilled to have been able to form with Jake Curtis and others the Ocean Biomedical as a way of trying to move discoveries at the bench quickly into the clinic. There would be nothing that would make me happier than to have our interventions, whether they be cancer-focused, fibrosis-focused, or infectious disease-focused, such as malaria, to get them into patients as quickly as we possibly can. I don't have to convince anybody who's listening in about the importance of cancer. You need to listen for 5 or 10 minutes to Dr. Curtis's discussions from our previous session when you realize that malaria is the #1 killer of children, and he has a vaccine that can potentially control all that. It's truly exciting to be in a situation where we have interventions that can control cancer, interventions that can control malaria, interventions that can control fibrosis, and that we have a chance now of moving them along as quickly as we can into new and effective therapeutics. I'd love nothing more than to have that as our legacy and the legacy of Ocean Biomedical.
Well said, well said. It is very impressive, and it's always been a core mission of this company that it's working on large unmet medical needs. And we can now put hepatitis B in there as well, chronic hepatitis B. And all these are diseases that affect global populations and that have a lot of room for new therapeutics. I know it's not an easy climb, but it seems to be a climb you enjoy doing.
It's been fun. It's been very interesting.
Oh, good. Well, thank you so much for your time today. I know this is extremely complex science. I think Dr. Elias has done a great job of helping put it in terms that at least most of us can understand. And I feel like it's very beneficial to do this from time to time, to talk about the science in this way.
Thank you, Kevin, for putting this together. And as always, we're available for questions that come up.
Let's do it again soon. Thanks, everybody, for tuning in. And if you do have specific science questions, you can send them to me at connect@oceanbiomedical.com. We'll try to pass them along to the scientists, and otherwise we will keep filling in gaps in the details on this as we can. Thanks so much for tuning in, and we hope to do it again.